Hello, and welcome to all the participants who have already logged on. We are excited to discuss our platform with you. We're just going to wait for a few more to join, and then we'll get started in a few minutes. Welcome to everyone. We'll just give it about one more minute. Okay, great. It is 1 o'clock. So thank you all for being with us today and welcome to our webinar. We are very excited to share with you our inflammatory disease mouse models that can help you identify novel therapeutics targeting cytokines and/or their cognate receptor. But before we get started, I would like to introduce myself and our expert panelists. I am Rebecca Soto, and I will be the moderator for today's webinar. I have a PhD in biological and biomedical sciences, and my primary role at Biocytogen is to develop scientific content. I'm joined today by our speaker, Dr. Jenna Frame. Jenna is the manager of science communication and marketing at Biocytogen and has over 15 years experience working with genetically modified animal models in the field of developmental hematopoiesis. I'm also happy to be joined by two expert panelists, Dr. Clark Gasper and Dr. Luke Yu. Clark is a senior business development manager at Biocytogen, and his previous research experience ranges from protein biochemistry and structural biology to antibody production and small molecule development. And lastly, Luke serves as our deputy general manager and global head of preclinical pharmacology here at Biocytogen. Luke has held several leadership positions, and specifically in the area of drug discovery and development at both the preclinical and clinical stage. And so with that, I'd like to welcome our speaker, Jenna, and our panelists and every one of y'all for being with us. And we will be taking questions at the end, so please stay tuned. So I would like to invite Jenna to begin her presentation.
Great. Thank you, Rebecca, for that introduction. I'm so excited that you all are joining us today, and I look forward to discussing, introducing these models to you. So let's get started. So before we dive into the disease models, I'll just start by providing a brief introduction to our company and the services that we provide. So BioCytogen was founded in 2008 in Worcester, Massachusetts. We initially focused on genetic engineering, where we developed proprietary methods that improve the efficiency of gene knock-in, as shown here, by 10 to 20-fold, even for very large insertions. So with this platform, we've been able to supply over 3,000 custom animal models for both academic and industrial clients worldwide. We've since then expanded our service platforms to include preclinical pharmacology and therapeutic antibody discovery, which if you look at sort of the timeline here of antibody drug development, these platforms utilize our humanized immunoglobulin mice platforms, RenMab, RenLite, and RenMab hits. So altogether, we've integrated these gene editing, animal model, antibody discovery, and pharmacology platforms to sort of seamlessly provide a platform to help you accelerate the entire process of drug discovery. So for the rest of today's presentation, I'll start with a brief overview of autoimmune disease initiation and progression. I'll then discuss how we can model these diseases in mice, including multiple sclerosis, rheumatoid arthritis, and colitis, each with specific case studies. And then finally, I'll mention additional models and tools that can help you to streamline your drug discovery workflow. So autoimmune diseases have complex pathophysiology and are thought to arise from the combination of genetic and epigenetic factors and various environmental triggers, such as infection, smoking, and diet. These initial triggers result in abnormal or defective immune responses that can cause a decrease in expression of self-antigens by the thymus. This leads to a reduced activation threshold for self-reactive lymphocytes, resulting in their defective inactivation or elimination. So over time, these self-reactive lymphocytes can persist and contribute high levels of autoantibodies and cytokine imbalances that over time eventually lead to disease. These specific cytokine imbalances resulting from autoreactivity really vary depending on the disease. However, as you can see, many of the inflammatory cytokines overlap between diseases that they're associated with, and these targets in particular have been of interest for discovery of new therapeutics. So in order to test these potential therapeutics in a clinical setting, BioCytogen offers disease models in various strains of humanized cytokine mice to help validate your drug candidates faster. So this brings us to our first poll question. So hopefully you can see that. So just want to ask the crowd, if you know the answer, which diseases could potentially be alleviated by IL-10 therapy? Okay, so yeah, it looks like we have most of the answers here. So yeah, it's exactly right. The answer is really all of these. So let's see. So IL-10 is really a classic anti-inflammatory cytokine. And so deficiencies in IL-10 or IL-10 receptor genes are in particular linked to susceptibility to colitis, but could potentially be used to treat these other inflammatory diseases as well. So the first disease that I'm going to introduce today is modeling multiple sclerosis. So as a brief overview to this disease, multiple sclerosis or MS is a chronic inflammatory disease of the CNS that leads to encephalitis and demyelination. MS is caused by autoreactive T cells and results in symptoms such as muscle rigidity, paralysis, visual disturbance and blindness, and sensory loss, as well as ataxia. Different types of MS can be characterized by the presence of relapse or progressive disease. And as you can see on the right side, here different strategies have been introduced to either inhibit neuroinflammation directly, such as mitochondrial protective strategies and remyelination strategies, as well as strategies that work to prevent the proliferation and migration of T cells to sites of injury. So at present, the experimental autoimmune encephalitis model, or EAE model, is widely used in the study of multiple sclerosis due to its pathological characteristics of inflammation and demyelination, just like MS. So in mice, as well as rats, EAE models can be induced by immunization with a variety of different protein or protein homogenates, spinal cord homogenates, including spinal cord homogenates, purified myelin, myelin proteins, or peptides of these proteins. Now for our studies, we use MOG, which is myelin oligodendrocyte glycoprotein. Typically, this is in an emulsion that promotes prolonged release of antigen, and the pertussis toxin, or PTX, is also given to the mice for sensitization. So what happens is after the injection, myelin-specific T cells are activated in the periphery and then can enter the CNS tissue, become reactivated. And this triggers a series of inflammatory responses. So in addition to this active EAE disease, there's also transgenic models that can be used, or adoptive EAE, which is induced by transferring T cells from active EAE mice. So in these models, typically demyelination can be less severe, and so sometimes they're stimulated by additional autoantibody fusion. But these two models can also be very helpful for mechanistic studies. So this brings us to our next poll question. So let's see if I can. So which of the following is not associated with murine EAE disease? Okay. Okay, so I'll just share those results. So yeah, the answer is C. So typically T cells are the primary cause of EAE disease. So I'll just move into description of the EAE model. So at Biocytogen, we really focus on the active EAE model for testing therapeutic interventions. So this study involves wild type mice that are 10 to 13 weeks old. As I just described previously, they're immunized with the MOG, the myelin oligodendrocyte protein emulsion, for prolonged antigen release and sensitized by PTX injections, as shown here. So when applicable, test article treatment is performed starting on day 10, which is around the time that symptoms begin to manifest. So in this first study, EAE was induced in wild type C57 Black 6 by immunization with MOG emulsion and PTX. So compared to the control, you can see that the MOG PTX treated mice, which is shown in red, displayed higher clinical scores, shown here. And these clinical scores really reflect things like tail weakness, lameness, high limb paralysis, and other symptoms that are kind of combined into this clinical score. As you can see, this disease also induces some weight loss due to the inflammation and other symptoms, as shown here on the left. So together, this data demonstrates successful induction of a stable EAE disease in the wild type mice. Histologic analysis can also be performed in mice with active EAE. So here at the endpoint of the disease, tissue sections were stained with H&E. And what you can see here is in the mice treated with a MOG to induce EAE, that there was more inflammatory cell infiltration in the spinal cord of these mice, shown here, compared to the vehicle control. And using immunofluorescence, you see that there's a qualitative decrease in the expression of the myelin basic protein, shown here in green, in the EAE-induced mice. So together, this data demonstrates, again, that successful EAE was induced in wild-type mice. Next, we sought to induce EAE in our humanized model that expresses human IL-17 in place of mouse IL-17. So again, similar to the results in wild-type mice, you can see that the MOG-treated mice have a significantly higher clinical score, shown in red, which is also reflected here in the weight fluctuations observed on the left side. In addition, similar pathologic changes were observed at day 45, including increased inflammatory cells in the immunized mice. And again, the myelin protein was significantly decreased in the treated humanized mice, as shown here in the inset. So this further demonstrates successful EAE induction, meaning that it is possible to induce EAE in humanized mice, so we can test the efficacy of novel anti-human cytokine therapeutics in this setting. To further assess the disease progression on a cellular level, we next isolated the lymph nodes from the humanized mice with EAE induction and stimulated T cells for 6 hours with PMA and ionomycin in the presence of profeldin A. So cytokine producing cells were then analyzed by FACS. So as you can see on the left side, the percentage of IL-17 positive, CD3, CD4 positive T cells was increased in response to MOG immunization. And so was the number of interferon gamma positive T cells shown here. And the overall cell count was shown here to increase with MOG induction. So taken together, these data indicate that the model could be useful to investigate new anti-human drugs to limit EAE disease progression in humanized mice. So the next disease model that I'll introduce is a rheumatoid arthritis model. So rheumatoid arthritis is among the most common autoimmune diseases, and its clinical symptoms mainly include generalized joint swelling, pain, and mobility issues. Severe cases of RA can lead to disability, as evidenced by hyperplastic synovitis, cartilage injury, and bone structure loss. First-line treatments for the disease include steroids, and for severe cases, biologics that block cytokine-induced inflammation. So pathogenesis of RA typically involves infiltration of inflammatory cells and autoreactive CD4 T cells into the joint tissue, resulting in pro-inflammatory cytokine production, complement activity, and autoantibody deposits. These cytokines that are produced induce production of MMP and rank ligands by fibroblasts that ultimately activate osteoclasts and destroy the tissue, resulting in joint destruction and pannus formation, which is a type of granulation tissue. So in the lab, collagen-induced arthritis is a common experimental model of rheumatoid arthritis, and this is induced by immunization with type 2 collagen, or C2, and this is the major constituent protein of articular cartilage. So animal models immunized with COL2 develop autoimmune polyarthritis, which shares several clinical and histologic features with RA. So in this set of studies, we used mice on the C57 Black 6 background. The immunization is given on day 0 and the challenge is at day 21, as shown on the chart. And the test articles are typically provided around onset of the disease and given every 3 to 4 days. So in this first proof of concept study, mice immunized with collagen-2, you can see that they gradually worsen clinically in terms of disease score, which reflects generalized joint swelling, pain, and mobility impairments. So these symptoms, as shown here on the right, are observed at about a 60% incidence in this strain. So the examination of joint pathology in these mice indicates inflammatory cell infiltration, and narrowing of the joint spaces and destruction of normal tissue architecture, compared to the wild type mice here, which have normal spacing, lack of stenosis, and lack of inflammatory cell infiltration. So together, this indicates that the disease was successfully established. So in this next case study, we wanted to test the efficacy of methotrexate, which is a first-line therapy for this disease. So in this experiment, the severity of the disease score, which is shown here, was reduced with administration of the drug, as shown by the green line here, and the incidence of symptoms was also significantly decreased. In addition, the methotrexate group experienced less weight loss when compared to the collagen-2 immunized group shown in red. The overall assessment of ankle joint pathology in these mice indicates that the mice treated with methotrexate, which is shown here on the right side, had less dramatic histologic changes. So while collagen-2 alone had significant disruption to the joint architecture, those immunized mice treated here had just some inflammatory cell infiltration and slight edema. But overall, these data, including the pathologic scores here, suggest that the RA disease not only has been successfully modeled, but also methotrexate treatment can show efficacy in this mouse model. So that brings us to our next poll question. So which of the following cytokines is not yet implicated in RA pathogenesis? Okay, so if you were hearing the first poll question, you may have guessed A, which is correct, the anti-inflammatory cytokine IL-10. However, these other pro-inflammatory cytokines do appear to play some role in disease progression and are currently targeted by some therapeutics. So in this next RA study, we aim to test the efficacy of anti-human IL-6 in this model. And before I introduce that study, I would first like to introduce our humanized IL-6 receptor model. So this model expresses mouse IL-6 in place, human IL-6 in place of the mouse counterpart, is shown here by ELISA. Likewise, this model expresses the human IL-6 receptor on the surface of TCR beta positive T cells, as shown here by flow cytometry. So in this study using humanized IL-6 mice, the anti-human IL-6 antibody sirukumab was given at 2 separate doses at the indicated time points shown here. So as you can see from the graph on the right-hand side, the disease was successfully induced in immunized mice, as shown by the disease score. And the severity of the disease in this experiment was also reflected in the body weight of these animals. However, as you can see, the disease score was significantly improved with the higher dose of the antibody therapy, as shown here by the blue line. So these results indicate that IL-6 blockade was able to improve arthritis-related clinical scores in these mice. Histologic examination of the joints by H&E staining indicates that, as previously shown, mice immunized for collagen-2 alone had significant damage to the bone structure and loss of periarticular spaces when compared to the control mice here on the left side. However, the immunized mice that received high doses of sirukumab had less severe pathology than col2 alone, exhibiting just partial pannus formation and overall reduced disease scores, as shown here by the blue bars. So together, these results indicate that blocking human IL-6 was able to improve the arthritis-related disease in these humanized mice. So the final disease model I'll be presenting today is a model of inflammatory bowel disease. So inflammatory bowel disease refers to chronic inflammation of the colon or GI tract and includes ulcerative colitis and Crohn's disease. Clinical symptoms of IBD include recurrent diarrhea, abdominal pain, intestinal bleeding, fever, and weight loss. Though the pathogenesis of IBD is not clear, studies have shown that various factors such as genetics, immune system, external environment, and intestinal microorganisms are associated with occurrence of the disease. Disease pathogenesis in IBD typically involves a breach of the mucus layers, bacterial entry, and immune system activation that ultimately results in cytokine imbalances and a chronic inflammatory state, resulting in expansion of the lamina propria. Current treatments for IBD typically involve steroids, or in steroid refractory patients, cyclosporin A to reduce cytokine production and lymphocyte function. Immunomodulators and anti-cytokine therapeutics are also utilized. So a variety of preclinical mouse models are currently used to study IBD. I'm going to introduce today the mouse enteritis model induced by dextran sulfate sodium, or DSS, because it's the most widely used chemically induced IBD model. So in this model, acute ulcerative colitis is induced by dissolving the DSS in the drinking water, which destroys the intestinal epithelium in the mice and results in cytokine release and inflammation to recapitulate the disease. Animal models of IBD exhibit clinical symptoms and pathological features similar to those present in human ulcerative colitis. And the criteria used to determine disease severity, which I've listed here, includes significant weight loss, alterations in fecal morphology, including presence of blood, as well as colon weight and length. And of course, we also perform pathologic analysis that allows the cellular assessment of inflammation and things like granulocyte infiltration. So as the first proof of concept study, we first assessed the efficacy of cyclosporin A at mitigating the effects of DSS-induced disease in these mice, and this is in wild-type mice. So as you can see from the upper graphs, mice treated with DSS alone, shown in green, had significant weight loss compared to the control mice. And this weight loss was really ameliorated in the mice treated with cyclosporin A, shown here in blue and red. In terms of the disease activity score, the mice treated with DSS alone had worsening disease over time, while the mice treated with cyclosporin A, as shown again by the blue and red, experienced less severe symptoms. Evaluation of the colons of these mice at the study endpoint indicated that mice treated with DSS alone had shorter colons with altered weight to length ratios, as shown by the green bars. However, the mice exposed to DSS and treated with cyclosporin A did not exhibit significantly different alterations in colon length and weight when compared to the control mice. So this indicates that CSA treatment helps to prevent the onset of these symptoms in the DSS-induced IBD mice. This is further evident by the histologic analysis of the colons of these mice. So as you can see, the morphology of the colon is significantly disrupted in mice treated with DSS alone. There's significant hyperplasia and thickening of the lamina propria and inflammatory cell infiltration. These phenotypes are significantly alleviated by treatment with cyclosporin A, and the changes are quantified here on the right-hand side in terms of the histopathology score. So altogether, these data indicate that DSS-induced IBD was successfully established in the wild-type C57 black cyst mice, and that cyclosporin A could significantly reduce the IBD-like lesions. In this next pilot study, we sought to intervene with anti-TNF therapy, which is the current treatment for IBD in humans. So in these experiments, both a preventative and therapeutic treatment was given for the antibodies either on day 0 or on day 3 and day 5. So as you can see from the graph, the therapeutic treatment of the mice shown by the green bars helped to reduce the weight loss induced by DSS. Examination of the colon length in these mice indicates that the anti-TNF treatment on day 3 and 5 post-DSS administration modestly but significantly improved the colon length compared with DSS alone, shown in blue. So further studies with this model in-house are currently ongoing. So that brings us to our final poll question. We would love to hear from you which model you're most interested in at this time. Great, looks like we've got a variety of interests in the audience. Okay. So in addition to the few humanized models I introduced throughout the presentation, I wanted to show a more comprehensive list of humanized cytokine models that are directly relevant to the diseases prevented today. So you can see that we have several models in the IL-6, IL-1, 3, 12, 17, 21, SPP1 for migration studies, and TNF families for you to explore your novel anti-cytokine therapies. And for a full list of our models, you can scan the QR code here to download the catalog from our website. Finally, I also want to mention that we have started building a tissue library as a companion to our humanized mouse catalog. So the library consists of tissues from various humanized mouse models as a way of providing samples to you for validation assays before having to commit to either a large order or arrange shipment of live mice. We currently have tissue available from these 5 models, but we are accepting requests from other models from our catalog as the library continues to grow. So if you're interested in one of these or another model, feel free to reach out to us and we'll be happy to help you out. In addition to the off-the-shelf models that we provide, if you don't see the exact model that you are looking for, just a reminder that we have an incredible gene editing team that offers comprehensive services for both models and cell lines using technologies including CRISPR, traditional ESC-based homologous recombination, and more advanced technologies as well, depending on your specific needs. And we offer some of the fastest timelines in the industry. Finally, I just want to introduce a more comprehensive list of the pharmacology services that we can provide in addition to the few that I mentioned today, including histology and in vivo analyses. We do offer several in vitro ex vivo offerings in addition to the modeling capabilities I shared earlier. Okay, so to summarize what I've told you today before we jump into the Q&A session, I hope today's material was able to demonstrate on a surface level how these models can recapitulate symptoms of autoimmune diseases for your preclinical work, despite complex etiologies of the human diseases. What these models can do for you is really enable MOA studies that can jumpstart your therapeutic drug discovery, but also be recapitulated in humanized mice if you're interested in testing the efficacy and safety of specific anti-human therapeutic interventions. So with that, I would like to thank you very much for your attention. You're always welcome to email us at info@biocytogen.com, and you can call us as well. And I'm happy to answer any questions right now in the Q&A session.
Yeah, so thanks, Jenna, for that wonderful presentation. It looks like we have some great questions in the chat and the Q&A. So I'll start first with, how do you determine the frequency of dosing with a test article?
So I can take that question. So usually our clients will have initial data that helps to inform the dosing of their specific test article. But if they're unsure, we can look through the literature for previous dosing schemes or suggest doing an initial dose response study or maximum tolerable dose before proceeding to efficacy.
In addition, I suggest to do the PK study first before you do any in vivo efficacy study. So then you get some good idea about which dose do you need, also frequency. So yeah, also if you want to get more information, definitely I agree with Clark. So you have different dose level for the animal study or dose range finding study.
Sounds great. Okay, so next question. Are you aware of any differences in the EAE model when using a B6J versus B6N wild type mouse? So I believe they're referring to the C57.
Maybe I can answer this question. So maybe in the most animal disease models, 6N and 6J may very similar, but in some disease model they did have some difference. Like we try to use 6J and 6N in the EAE model. I think based on our experience, so 6J sounds like the disease model is more stable. Yeah, so 6N maybe a little variation, but I think they do have some difference in this model.
Okay. Next question, what parameters are involved in the calculation of clinical score in the EAE model?
Clark, do you want to answer or I can answer?
Yeah, so I don't have that offhand.
Maybe I can answer because basically clinical score is really by a clinical observation. So we can, that's a pretty standard protocol for the EAE model. So basically we just observe like a tail weakness. Maybe if it's normal, is like we score 0. If we score 1, the tail have some weakness that should have. Also, the second, the tail cannot have some reflex to some stimulate, and also some hind leg maybe have some paralyzed. That's a score 3. You see, just 2 hind legs have totally paralyzed. That will be score for 5. That's the highest score. Or if an animal death, that will be highest score. So totally by the clinical observation.
Okay, great. Next question is, in my experience, the CIA model works better in DBA mice. Is it the same for you? And are your humanized IL-6 mice BL6 or DBA? And if they are BL6, why did you choose this background? So I do know that the IL-6 mice are in a BL6 background, but if y'all would like to add to that.
Definitely the DBA is a good strain, a proper strain for the CIA model. But we choose the B6 because most of the humanized mice, they use the B6 as a background. So that's why we choose the B6 for the CIA model. CIA model for the disease progression, definitely the severity of the incidence of the CIA in the B6 program may be lower than DBA. So definitely DBA, the disease severity or disease progress is more stable, also has a high incidence rate than B6. But since most of humanized mice in the B6 background, so we chose the B6. But a lot of disease model, arthritis disease model, we call the CAIA. There's a collagen antibody induced arthritis. We also have this model available. This model can be used for all the animal strains, including the B6, also with very consistent disease progression. And so that's similar to DBA.
Okay, that's great information. Next question is, does CRISPR-mediated knock-in occur in zygotes or ES cells?
I'm not an expert in this area, but I think we can consult our gene targeting team. They have a lot of good experience to answer this question.
Right. So yeah, if anyone's interested, please feel free to reach out. Could you please discuss atopic dermatitis models you have available at Biocytogen, and have you tested anti-OX40 or the ligand antibody treatment in these models?
So for atopic dermatitis models, currently we have an oxazolone-induced model that we use in either wild-type black 6 mice or our double humanized IL-4 mice, I believe. And so we have some validation data that we produced, and so we are happy to follow up and provide those as examples.
Yeah, I'd just like to add that if you missed our previous webinar, you can watch that. Also, we have some information on our website in case you're interested in the AD mouse model. So for EAE induction, you mentioned MOG emulsion. Can you explain what it is and the mechanism?
Yeah, so as Jenna mentioned, MOG is myelin oligodendrocyte glycoprotein, and this is a transmembrane glycoprotein that's expressed on the surface of myelin membranes. And so it's been proposed that the presence of MOG at high levels leads to production of autoantibodies against neuronal cell adhesion proteins. And so this pathway leads to the demyelination of the neurons that causes the disease.
Perfect. So we have another related question about the C57BL6 model. Why was that used instead of DBA for the CIA model? I think Luke answered it perfectly, that our humanized mice are in the C57BL6 background, and so that's mainly why, for consistency purposes. So the next question is, can any other EAE stimulants be used besides MOG, since many demyelinating conditions are not MOG positive?
Yeah, absolutely. As I briefly mentioned, there's all types of different ways to induce EAE, really depending on what type of readout you're looking for.
Great. And so several of the models you showed included histology as part of the readout. Do you recommend histology analysis as part of disease modeling studies?
Yes, so it would depend on the specific disease, but most of these diseases result in tissue damage or immune cell infiltration that can't be observed very well externally. And so for cases like this, we recommend histology, like with the EAE or IBD models, just to see how extensive the disease groups differ from control or treatment groups.
Okay, great. So next question. There's a lot of great questions. Do you have cytokine profiling data for each model?
So at the moment, we don't have that cytokine for each model, but we do have the capability to produce that. We mainly use the Luminex platform in order to multiplex cytokine analysis.
Perfect. So why do you think you saw better efficacy of the therapeutic treatment with anti-TNF in the DSS model in comparison to starting treatment at day 0?
Yeah, I mean, so for that small pilot study, it could be that there were 2 treatments in the therapeutic window as opposed to 1 preventative treatment. I don't know if Luke, you want to add on to that?
I think because there's a preventive treatment just a single dose, right?
Right.
So there's there's small molecule treatment that may have a short half-life, you may not, not show better efficacy. For the therapeutic treatment, there's 2 doses. So I think small molecule maybe they can have some constant activity in the animals. So that's why they show the better efficacy for the therapeutic treatment. Also, maybe in this time period, maybe for more related to some cytokine releasing, timing zone. I think that could be the reason.
Okay, great. So how does the CA model with collagen and LPS stand against the collagen model?
LP, CAIA stand against the collagen 2 model.
I mean, can you say again? What's the question?
The CAIA model with collagen and LPS stimulation.
Right.
I don't know if we've done a head-to-head of those two models.
Yeah, we did because yes, that's the CIA model. When we give the antibody, collagen antibody to the animals, we also use the LPS to boost the disease progression. That's so that's very standard protocol. So, and also this as I said, for CAIA model, there's a more like a free about 100% incidence. Also, so they have a very short disease progression period. You know, that's a really because the disease progression is related to the antibody titer. So basically, right after LPS stimulation, so you can start the treatment. The disease will come out. So maybe after day 3 or day 2 after LPS treatment.
Okay, so much faster.
Okay, so have y'all developed inflammatory cell death models such as for ferroptosis?
That's in which model? Right, that's very general question, right?
Right, yeah, that's
Yeah, we do have capability to test the apoptosis.
Can your team perform detailed cytokine profiling in these models? I think Clark alluded to that earlier, that we definitely have the capabilities to do that. Do you have an IBD model that presents with fibrosis phenotype in the gut?
Yeah, we can test it by histology. That's pretty easy to do.
Can you see the relapse/recovery phenotype from score 2 to score 1 or 3 to 2 in the EAE model?
Yeah, we did not just specifically to see this kind of recovery in this disease model, because sometimes maybe yeah, some days maybe after treatment it may just have a reduced score. We can observe in our animal model. Maybe that's sort of a recovery.
Okay, what is the advantage of using humanized mice for the study of these diseases over other wild-type mice?
I think that's in the single that's an improved translation, because we use a humanized antibody because for in the clinic, so it's better to use the same type of antibody in the animal study. They can improve the translation.
Yeah, it would also depend on your test article. If it targets like a human receptor or protein, the humanized model would be the way to go.
Yes. How can we distinguish inflammatory disease versus autoimmune disease models?
I think that major differences between inflammatory disease and the autoimmune disease is about autoantibody generation, right? Because for the inflammatory disease, basically they may trigger by some inflammatory cells or cytokines, but for autoimmune disease, they will trigger some B cell activation, generate autoantibody. I think that's a major difference.
Yeah, like for asthma, for instance, it would be an external allergen versus an internal auto reactivity.
Right.
Yes. What is the site of activation and proliferation of T cells in EAE mice? Is it the spleen or spinal cord?
Yeah, so previous studies have looked at MOG reactivity of both CD4 positive and CD8 positive T cells. and those from the CNS were associated with increasing disease severity. And so it suggests that the cells from the CNS and not the spleen are involved in EAE development.
Also, in addition to that, so the immune response in the EAE model is systemic, not only in the local. So basically, the cell, dendritic cell or activated to T cells, T cell activated activate T cells can penetrate through the BBB to the mining site to trigger the mining injury. So this T cell proliferation can see in both systemically, maybe also in the local in the spinal cord. So you can definitely consider some T cell activation in the spleen.
Right. Okay, so have you developed models for lupus? And if so, what would be the best readouts?
For lupus, basically we normally, most people, we just use a spontaneously lupus model like NZBW and MRL mice for the lupus study. We also we call the lupus nephritis models. But also in our company, we can use some compound to induce lupus-like disease. So right now is in the development. So I think soon we're going to have this induction model of the lupus. So we'll give you an update soon.
Sounds good. And so we have another question about what about the inflammaging? Is there a model we could be able to use to assess inflammation due to aging rather than just using old mice?
Can you repeat the question? Maybe I didn't follow both of the question. Exactly.
Yeah, so they want to study inflammatory disease associated to aging, but they want to know if there's models to kind of study this rather than just using old mice.
I think you take it either way. We just use older mice. So actually, in our company in Biocytogen, we'll have a lot of aged mice, so they could be used for this kind of a study.
Yeah, so no specific.
Yeah, not at this time.
Yeah. Okay, so we have a few more in the chat. Will antibodies have access to the central nervous system?
Simple answer is no.
Right.
Yeah, so usually if the CNS is intact, no. But if there's the barrier is compromised, then they can.
Okay, so there's always a debate on targeting the ligand or the receptor. And so from an antagonistic point of view, which is the preferred target, receptor or ligand, and any feedback in cytokine production after the blockage of cytokine in general?
That's a great question, because what we discussed about is which one is better to be blocked, like is ligand or receptor. But put it this way, you say there's a one ligand, can target the multiple receptor. Maybe is a good way to block it, maybe the ligand. But sometimes the one receptor can bind to multiple target ligands. So that really depends on the target. Sometimes maybe one drug like block one ligand, we show some efficacy. Same thing, another company just developed a different drug to target the receptor. So maybe they have the pros and cons, but it really depends on the biology of the target.
Okay, so what is the site of activation and proliferation of T cells in EAE mice, spleen or spinal cord?
I think we answered that.
Yeah, we answered that one earlier. How does the CAIA model with collagen antibody and okay, yes, that
Yep.
was in the Q&A earlier. Sorry. Is there any animal model specific to alpha 4 beta 7 study in IBD?
Or beta 7.
I don't think we use this IBD model, use this humanized mice in the IBD model, but if you are interested, we can definitely to develop this model in this humanized mice.
Okay, so would you be able to access inflammatory cytokines using tail blood draws, or would these cytokines be too localized?
Yeah, they should be careful to do it.
Yeah, it would depend on the extent of the disease induction. So just one example is with atopic dermatitis, we use 2 sites. One is the back and one is the ear. If you were to use only the ear, you don't get systemic cytokine induction. And so again, it depends on the disease severity.
Okay, great. And we'll just take one final question. So does the immune profile similar to the normal wild type mice in the cytokine TG mice?
Yeah, I think we have this kind of comparison, right? That's very similar, but it does depend. If you say there's a humanized mice for these specific cytokines, the level sometimes that's comparable to the physiological level of the mice in the mice, right? But in some humanized mice, maybe a little different. But as long as they can certainly have achieved the level to show some physiological activity.
Okay, sounds great. Well, thank you everyone for your wonderful questions, and to our panelists today. I would just like to remind y'all that you can contact us via email, phone, or in the contact page located on our website if you would like to discuss any of our models and services. We would really love to partner with y'all. And if you have any additional questions or wanted further elaboration from some that were asked, please, again, feel free to reach out. But thank you all for being here today.
Yep.
Thank you, Rebecca.
Thank you.
Thank you.